So it's a great pleasure to introduce Professor Christoph Koch today, who's joining us from Seattle. Christoph was mentioned extensively yesterday, both in connection to his pioneering work on the biophysics of computation and the very beginnings of the entire field of computational neuroscience. As a matter of fact, I think we should credit Christoph with the beginning of our summer course, because we were inspired by a similar course that he created also in Woods Hole at NBL on computational neuroscience. He was also mentioned yesterday by Tommy in connection to his extensive and seminal work on consciousness. So Christoph is a true Renaissance man who has shed light on all kinds of problems in neuroscience and systems neuroscience and computational neuroscience. And it's a great privilege to have him here talking about the relationship between structure and function in the visual system. So Christoph, all yours.
Thank you, Gabriel. I was also the doctor father, as we say, the mentor of Gabriel when he got his PhD at Caltech. All right, so today I'm going to talk not about biophysics, about our consciousness, but about relating function and structure in the visual system of the mouse. This is all work done over the last 7 years at the Allen Institute here in Seattle. The vision we've been pursuing here since I left Caltech and joined the Allen is to do something similar to what astronomy has done. To build a set of instruments we call the Allen Brain Observatory that are similar to the large-scale astronomical observatories, where you can routinely record at high resolution data on a standard protocol and make all this data accessible, where people ultimately will apply. I want to do this particular experiment. We do the experiment and we make all the data available. And I'll come back to that at the end. So I'll talk to you about that. I'll tell you about that. 2 pipelines with 2 very large-scale instruments. One is optical physiology using 2-photon calcium imaging, and the other one is doing electrical recordings using neural pixel, where in both cases we now can record from 10, 50, or 100,000 neurons under standard condition, download all the data, make all the data available to anyone, such as you. So if you go to the link in this talk, you can see there's a link there to a data website where, using a standard Allen SDK, you can download right now. There's no login required, no password, nothing. You can download all the data and all the metadata about all the animals, everything that was shown, and you can analyze it. Of course, the vast majority of this spiking activity has never been seen by human eye. It's because there's so much. It's several billion action potentials, 100,000 spikes for 3 hours total. So in order to do this, in order to make this highly reproducible, we need a few, we need to engineer an entire ecosystem around this. So for once, we need to have a high resolution 3-dimensional coordinate system. We worked on this for the past 10 years. It's now sort of the standard coordinate system for the mouse. Of course, the mouse has some advantages over the human because we use inbred mice, C57 black 6J mice, which is sort of standard in the field. So they are highly genetically, they're all from the same background, unlike 2 different people. And then you can see there in this picture, this is superimposed an image, a horizontal cut through 1,500 mouse brain, 1,500 all superimposed because you can tightly align them. And so we have this true 3-dimensional atlas. Each pixel is 10 by 10 by 10 microns. There are roughly 500 million of them. We have 43 cortical areas in one side, of course, a total of more than 300 subcortical areas and white matter structures. And so everything we do, every time we record, whether it's patching from an electrode to get its transcriptome, or electrical recording or optical recording, we always know within very high resolution where that particular neuron is. We also need to build a high throughput pipeline where we take animals, here various transgenic animals, for instance. We do standard surgery, always using the same, we use robots to do this. We do intrinsic imaging, think of that like fMRI. So we know for each mouse we do, We know precisely where is, let's say, the primary visual cortex, where is higher order visual cortex. We do training under standard in standard boxes where we track the training history of every animal. Then we either do optical imaging or neural pixels. Then we do afterwards, we go back and do intrinsic imaging, and then we sacrifice the animal and do histology of its entire brain. So we know exactly, we can identify, for example, where the tracks were of the microelectrodes, or where, if we have particular fluorescent neurons, where they are located. We have this pipeline of standard surgery, intrinsic imaging, either this multiplane, multiphoton imaging on neural pixels, and then this serial 2-photon tomography for reconstructing the animal. We've built this high resolution cross-platform registration, so we can go back for optical imaging, 6 times, 10 times, 12 times. We can go back and image the same cells. We have a standard sort of way to present visual stimuli. So here, you can, for example, you can see in different animals, these are different tree lines. In each animal, we do this imaging, and then we have the standard way of mapping. This is the back of the brain, this is primary visual cortex, these are higher order visual areas. So we can map, so we know when we did optical imaging or when we put neural pixels in here, we know exactly where we were, both functionally, as identified by these intrinsic imaging, as well as structurally, because we do this histology at the end. So here you can see, this is a mouse running past a movie, a wonderful movie called Touch of Evil. And this is 2-photon imaging. And we track, of course, we have cameras, and we track the movement of the animal and its eyes, et cetera, et cetera. And all of this is available. So we did this over the first survey. We did large-scale survey of 60,000 neurons. I'm not going to talk about, here you can just see a snapshot of the way different neurons, here are 4 particular neurons respond to different static gratings, drifting gratings, natural scenes, receptor fields, movies, etc. And we work with the community to come up with a standard and an ecosystem called NWB, Neurodata Without Borders. So it's a large-scale project that more and more people are using now to represent cellular data, time series of cellular data, whether that's optical physiology or electrophysiology. And it looks like it may evolve into standard for the entire field, which would be a great boon to everyone. And then we also co-developed over the last 5 years, this is a consortium together with Genelia, particularly Tim Harris, he was really the chief engineer, and then UCL in London, the Wellcome Foundation, where we developed these high-resolution probes called neural pixels. So what they are, they're roughly they're 10 millimeter long, covered by titanium nitride, very high precision manufacturing using CMOS, where we have 960 electrodes, and you can select 384 out of them. They're spaced 20 microns apart. We're now going to even higher resolution, so-called neural pixel ultra, that are spaced 6 microns apart. But everything I'm talking to you about today is 20 microns apart. So you have a stick of silicon, machine silicon, 10 millimeter long, and you put it into the brain of a mouse. We've also developed versions 48 millimeter long for the monkey using the same base station. So this is the shank, 1 centimeter. This is the base. And then here in the base, you have all the electronics for amplification, filtering, and multiplexing. So all that you get here is a flex cable. You have 4 wires going out to a central board. Low noise in tissue, actually, it's 7 microvolt. In water, in salt water, it's more like 5 microvolts. Very, you can see very clean impedances. Fantastic technology, has really made a huge difference, because you can now record from this stick, you can record 100 or 200 neurons, and if you put 6 or 8 or 10 of them into one animal, you can in a single experiment record 1,000 units. Okay, and then again, there needs to be, so we have these neural pixel workstations, where we put under standard conditions 6 neural pixels inside the into the brain. Minimal damage because these are so small. They are 22 microns thick, 70 microns across, 10 millimeter long. Really, it's if you look at the histology, it's really very clean. Okay, so that's sort of the background. Now, over the last 10 years, we've also worked at trying to get the structure of the animal. So this project involves mapping function on structure. So the structure, what we've done early on, already quite a while ago here, where we mapped in a very systematic way in animals where we injected, so here in 1 out of 500 animals, we injected either in a wild type or a transgenic in a particular subset of animals. We injected in a particular subtype of cell, we injected some fluorescent tracer, anterior growth tracer, and then we can reconstruct the terminals, the presynaptic terminals throughout the entire brain, ipsilateral and contralateral, in all these different structures in the CCF. So these are, you know, several hundred structures. And so here you can see this log representation of the connectivity. More recently, we published this last year, we've done, again, anterior growth viral tracing with Cre-dependent viruses in 1,200 animals in distinct mouse lines, where we can look at the pattern of termination. So here we have somewhere in cortex, goes somewhere else in cortex, and you can see it either you know, terminates in the superficial layer, only top of layer 1, or mainly in, you know, very heavily in most layers except layer 4. And you can see all these distinct patterns here from a cortical source and here from a thalamic source. Then we used unsupervised hierarchical clustering of all these different experiments to identify 9 types of feed-forward and feedback connections throughout the entire brain. This is not just the visual system, this entire brain. And then we can arrange a hierarchy for that involves all 43, or in this case, a subset, 37 cortical areas and 24 thalamic nucleus in a very systematic data-driven manner. And we can assign a hierarchy score. So the hierarchy score, think if you have a perfect hierarchy, a one-step hierarchy, that hierarchy score would be, let's say, from a LGN to a simple cell in a classical tubule in vivo. That's a hierarchy score of 1. Then if you go from the simple cell in a simple step to a complex cell, that would be a hierarchy score of 2. So you can see the total depth of this hierarchy is only 2 steps in the entire mouse. Why? It seems, so it's a shallow hierarchy compared to, for instance, if you look at the Fellman van Esten macaque monkey hierarchy. Well, A, it's a small animal. It's, you know, it fits comfortably into a sugar cube under a cubic centimeter. B, cortex is very heavily connected. Almost, you know, more than 30% of all connections are actually there. But of course, the connection strength is log normal distributed. So there are lots and lots and lots of parallel of other connections. So this is what it is. Okay, with V1 here at the bottom, and sort of the orbital frontal areas and some thalamic areas at the top of the hierarchy. Here you can actually see the, this is just for the visual system, and this is for the entire brain. All right, so now let's try to replicate that structural, the anatomical connectivity. Can we also find it in the pattern of spikes, particularly as the animal looks at something, you have this wave of spikes that comes through the LGN into cortex. And so we've done that over the last 5 years. We put this paper out on bioRxiv last year, and it's now making its this paper, and I'll show you mainly figures from this paper. It's making its way through peer review. So here we have on the order of 100,000 units, all registered to CCF. So every cell after, so there's of course a huge amount of post-processing involved using, in this case, KiloCluster 2. We have this automatic way, particularly Joseph Siegel, the author of OpenEFIS, he's the first author, and he did all of this, where we have, for each unit, we have 12 different criteria by which we do spike sorting and quality control. And all of that data is also associated. That's also associated metadata. So for each unit, you can see what happens if I'm more stricter, if I do a smaller exclusion criteria, or if I change my exclusion criteria, and you can then go back and analyze everything. All of that is made available here using SDK. Okay, so this is a setup. So we have blue is cortex. These other is other areas. So we record, of course, because these are 10 millimeter long, and the brain from bottom to top, as I said, roughly, fits comfortably into sugar cubes. So it is under 10 millimeters. And so what we do in these recordings, we can record from each one of these cortical areas all matching receptor fields, but we can also record from the LGN, this is really the lateral posterior. Think of this as the pulvinar. In a monkey, this would be called the pulvinar, the colliculus. And in between here, of course, there are other areas like the hippocampus. All of that is recorded. So just to show you the difference between doing optical recording and electrical recording, we do both, of course. Optical recording, we record at 30 Hertz. We record the fluorescent signals. We don't record individual action potentials. We know, in fact, now exactly what we're recording because we're also doing simultaneously electrical patch recording and optical recording. But in general, what you're recording optically, you're recording in a plane, typically cortex, because the photons don't go much deeper than layer 5. We're recording in a horizontal plane, genetically identified neurons. That's a big advantage, fluorescent, the signals. The other advantage is we can track this over multiple days. So we can come back here next day or next week or even next month and record from the same cell at 30 Hertz. Electrophysiology, we're recording throughout the entire brain because, we can record, we have this long 10 millimeter stick, that's CA1, dental gyrus, CS3 of the hippocampus, LGN. We can go all the way down here to the hypothalamus. So electrical recording, we're recording at 30,000 Hertz rather than 30 Hertz. So it's 1,000 times faster. And we're recording spikes. And of course, spikes have some ontological standing that, fluorescent signals don't really have. The drawback is we don't know what we're recording from in general. We can distinguish, of course, fast spiking interneurons from regular spiking cells. But other than that, we don't know unless we do optotagging. We don't know the genetic identity of these cells. And we can come back the next day and record from this very same layer 6 neuron in AL. So each technique, like always in science, has advantages and disadvantages. There's no best technique. Okay, so you get these, what you get in this case here, you get 1,000 neurons in one experiment, and you can look at this now for 2 hours or 3 hours, up to 3 hours of experiment. As the animal runs or sits and runs past movies and gratings and flashes and whatever. So that's pretty cool, pretty unprecedented. Okay, so you can see, we're recording from many animals here. Typically, we're recording from 6 areas, sometimes up to 8. It depends exactly on the exact angle. And of course, we know all of that because after we sacrifice the animal. In each case, we use optical tomography to reconstruct the track and to place the electrode and then every unit into the CCF. And so we can study, we know the majority of cells in visual cortex, respond. So this is one particular cell. As we show as the animal runs past Gabor patches, full field flashes from gray to white or gray to black, drifting gratings, dot motion, static racing, natural scenes, and movies. And you can look at, temporal frequency as a function of orientation. You can look at different orientations. You can look at the flash, the timing when you have a light flash or dark flash. You can do classical receptor fields, and we do all of that in parallel across entire visual field. And so you can get here, for instance, this is a fraction of cells where we can use this procedure to automatically determine receptor fields. And so you can see, certainly early on, these regions have 70% plus receptor fields. Okay, yeah, and interesting, if you look at hippocampus, because as I said, these electrodes go from visual cortex through hippocampus through LGN, as a control, you can see, you know, they don't have visual receptor field, and you wouldn't expect that. Okay, so this is the figure that we spent many years to derive. This relates structure and function. So I showed you the hierarchy of last year's Nature paper, Julie, Harris, and Mihalas et al., where we look at using this unsupervised clustering in 1,500 mice, we look at the connectivity just within the visual system. So this is LGN, V1, these are different visual cortical areas, LM, RL, LP, AL, PM, and AM. Sorry, this is actually a thalamic nucleus. So the two thalamic nucleus is LGN and LP. As I said, that's like the pulvina. These other areas are all cortical areas. This is the feed forward, and these are all the feedback connections. So this is the structure, and we can precisely assign to each of these stages an anatomically hierarchy score. So now, how can we do the same thing for the electrophysiology? Well, so let's say here we have two units in and the units here are, I forgot, they're between two visual areas, and we look at the cross correlation in response to drifting grating. And what you can see here, this is it's shuffled, so it's shuffle corrected, it's jitter corrected in the cross and correlogram, and we have this sharp peak here with offset of 3 milliseconds. In other words, the unit in V1, this is a V1 unit. Well, these are two units between V1 and LM, a higher order visual areas, V1 and LM. And you can see the V1, the firing of the V1 unit follows 3 milliseconds later is followed by the firing of the LM unit. And so we look at, for such sharp peaks, we have a criteria for what a sharp peak is. I think it's 7 times above baseline. Within a window of plus minus 10 milliseconds. So that's a sharp peak. So when you have something within this time here that's very sharp, we count that, and we look at the offset. So here you can see the distribution of these time lags between V1 and LM cell. The median here is 4 milliseconds or something like that. And then what we can do, so here, this is the anatomical hierarchy score difference. So here what we do, we take the anatomical one, and for each case we look at, well, between LGN and V1, what is the score difference, right, between here and here. And we plot this here in this two-dimensional diagram. And then here we do the same, but now we do it using this functional criteria. We do the median cross-correlogram time lag, you know, sort of these time lag here across thousands of units, and we get this structure. And then we can correlate. We can say, well, what is the anatomical difference here between, let's say, PM and RL, and what is the time lag between PM and RL, and let's compute the cross correlation between those two. So the anatomical hierarchy score difference and the median time lag. And you get this very nice, very, you know, with very high statistically, certainly we get this very nice relationship using both Spearman and Parson and correlation and coefficient. So in other words, the bigger the anatomical hierarchy difference, the bigger the difference here between, let's say, here and here versus here and here, the bigger the average time lag in the cross correlogram, which makes sense. Now we also looking at other measures here. So we are looking at we're looking at 4 different measures and see how these measures change across the anatomical hierarchy. So here we're looking at the time to first spike. We compute for each of these different areas because we have this great thing. We can have in one animal hundreds of cells shown always to the same stimuli. So in many, many animals. So we can collect all of that data and show, for example, this is the response to an an on flash, full field flash to the LGN, okay, it's of course the first, then this is to, you can see these different areas here, okay. Here here in B, we look at the distribution for each of those thousands of neurons. We're looking for the distribution of first spike response time in response to this flash. Okay, so we have a criteria for determining what the first spike is, and we do that for all our neurons, and we plot those distributions. And then we look here the correlation between mean time to first spike and hierarchy score. And again, we get this beautiful correlation, very strong correlation between the anatomical hierarchy score and the first time to spike in response to full flash. Here we have an automatic way of, as I mentioned to you, of determining receptor field. And here we automatically determine the receptor field size. So for example, here we have LGN, they're smaller, somewhat smaller than in a higher order area called AM. That's almost at the top of the hierarchy. Here we superimpose all of the receptor field size. Here we look at the distribution of them across all the units. Here's, by the way, all the numbers of units that we have. So it's, you know, that's 5,500 units just in V1 alone. And again, here, so if you have these, you know, these sort of numbers, you get these very tight statistical relationship, beautiful statistical relationship, which of course a lot of jitter. Right? If you look at here the distribution, yes, so even in V1, you can get some cells that have 1,000 square degrees, but you know the average V1 is around 400. By the way, these numbers are large because we're dealing with a mouse here. This is not a primate like a monkey or human that has a high resolution fovea. Anyhow, if you look at this, you get these very tight relationships, you know, with these, you know, beautiful correlations. Here we look at to what extent units are phase locked, to what extent units are really like a simple cells, where they should be completely phase locked, or to what extent the cell behaves more like a complex cells. So we have this modulation index that really measures the difference in power of the visually evoked response at the unit's preferred stimulus frequency versus the average power spectrum. So this is the number, and then we take the log of this, and where small numbers indicate sort of where large numbers indicate sort of simple cells, and small numbers indicate, particular negative numbers indicate complex cells, because you expect they're early on to be simple cells and later on complex cells. And again, if we do that, you can see some here that, you know, that where they have really bimodal distribution, different areas. Again, you can see this beautiful relationship. Early on, it's primarily simple cells. Later on, it's primarily complex cells. And then here we are pursuing something, an idea C.J. Wang has expressed many times in his paper. Particularly, I'm thinking of Chaudhry et al., where we try to measure the time scale of neural activity. So we first assess the intrinsic time scale of each neuron across all the different brain regions we record from by fitting an exponential decay function to each unit spike count autocorrelation during spontaneous activity. In this case, when there is gray screen, nothing particular present in the environment, and we look at we compute the autocorrelation, and we look at the exponential decay. And here, the mean intrinsic time scale is not correlated with visual hierarchy at all. Oh yeah, I should have shown this. I haven't shown this. I just realized it's not correlated. So intrinsically, each neuron is not. It's not true that intrinsically a neuron in a higher area has a slower time scale, intrinsic time scale than a neuron, let's say in V1. However, if we look at the response decay time to visual stimuli, which we quantify here by fitting an exponential decay function to the autocorrelation during a full field flash. So here you can see some fits. Here you can see the distribution to of the autocorrelation time scale of all the units. And again, here you can see the correlation. So it's certainly true that as you go up the anatomical hierarchy, the time scale, the evoked response time scale certainly increases across the hierarchy, which is what C.J. Wang has shown in his beautiful models in his neuron paper. So as you go up the hierarchy, there's a strong correlation between mean response decay and anatomical hierarchy score. And we also looked at active behaviors. In fact, we're doing this more and more. So here you can see the mouse is running. And in this case, it's getting rewarded. When it correctly detects, you can see sometimes the image change, right? Same, 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 same. There, it changes. And now it's same, 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 same. Sometimes we leave out a flash. So it's a really interesting paradigm. It really relates to an odd man out paradigm, what people have done, right, to compute P300, P300B, things like that. But here done in a mouse. So now we can look at what happens in this behavior. So this is the paradigm. Each image is on for 250 milliseconds. These are natural scenes. And then 500 millisecond blank. So here you can see at level of LGN, this is when the image changes from A to B, V1 and area AM. This is just one exemplar units of here. I'm sorry, of these units right now, we have, it's a smaller number of animals. We're still complete, this survey hasn't been completed yet, but in these animals still, 1,000 units, 800 units. So here, for example, you can see that this is for this LGN cell. This is for the same stimulus, when the same stimulus is shown again and again. And this is when, remember, the stimulus is on for 250 milliseconds. And this is when the image is changed. And we're looking at a change modulation index, which is the difference of these two over their sum. So the CMI, this change modulation index, is always between -1 and +1. If the cell responds stronger to the changed image than to the same one, then it'll be positive. Between 0 and 1. 0, they respond exactly the same. +1, the response is much bigger. The larger the CMI, the larger the response to the change. And here it's in V1, the response is bigger, and here's in AM. And typically you get this, first you get this initial transient, and then you get a sustained response. This is very typical. Okay, so here you can see very nice, again, correlation. This is always across the anatomical hierarchy score from LGN down here to this highest visual area, AM, up here. This is time to first spike now for this active natural scene presentation. And just like before, when we looked at the correlation between the anatomical hierarchy score and the passive presentation of the flashes, here, under active condition, we get very similar, in fact, they're highly correlated, those two measures at a single cell level. But here again, we get this beautiful correlation between time to first spike and position in the hierarchy. And here also the change modulation index, which is always positive, goes up, highly significant. This is under the active condition. So this is when the animal has to perform the task. Here we're doing a replay. So the animal gets to see exactly the same sequence, the same timing as here. The only difference is here the animal is not being rewarded and it's just running, while here it's actively being it's being rewarded. So you can see the difference that this active behavior makes. So LGN, for instance, there's no difference. LGN, whether the animal is in an active or passive phase, is the same. But as you go to higher areas, the difference grows. Now we can also do decoding, and you can decode from many areas, here we ask the question, we always take 20, we have a random forest decoder and we look at 20 units in LGN or V1 or LP or AM, and we ask, can we decode, how well can we decode the stimulus? And we can decode it to some degree. And then, so effectively, we find that spiking activity in any area, including very early areas like LGN, can be used to decode image changes with significantly above chance performance, although in LGN it doesn't do nearly as well as high up in cortex. Now, what we do, we assess how well the decoder performance correlates with the actual behavioral choice of the mouse in each trial, right? We can say, using my random forest decoder from 20 neurons on this particular trial, what did my decoder decide what the stimulus was? And what actually did the animal decide? Did it decide it was the same or different? And then I can correlate that, right? How well does the decoder correlate with the animal performance of the animal on that trial? And this very steeply correlates with anatomical hierarchy. It goes to almost 90% once we record in the midbrain reticular formation or some of the premotor nucleus. Of course, you would expect that. It's 0.25 in the highest area that we record from the highest visual area, and goes down basically to chance at the level of the LGN. So at the level of the LGN, although a random forest decoder can, with some above chance probability, decode the identity, it doesn't relate at all to what the animal does. So this tells us the animal doesn't access LGN. And so again, this is the difference. You have to be careful with inferring correlation just because you can decode something. So this is, rather nice, satisfactory result. And of course, this increase in change-related signals suggests that unexpected stimuli, right? This is really an odd man out paradigm. Unexpected stimuli are amplified by successive levels of the hierarchy. And this is consistent with some other oddball paradigm in the rat. But they're also compatible with general theories of hierarchical predictive processing, which posit that unexpected signals are preferentially passed on to higher processing stages. All right, so my last two slides, I wanted to come back to this open scope, what we call open scope, because we're now moving towards this, where we want to do the following. We want you, the entire community worldwide, to come up with a clever experiment. In principle, what we're working on with others over the next 3 years, that it can be in any part of the brain. Let's say you want to do an experiment, maybe a modification of this paradigm, maybe a go, no go paradigm. Using some particular, let's say visual, there has to be some restriction, let's say maybe a visual or maybe an auditory paradigm. You submit your proposal to us together with a bunch of other proposals. We, together with the external community, maybe you've done some piloting, so you have some idea this sort of can work. You have a particular hypothesis. You submit your experimental protocol to our request for proposal. We review these based on how interesting they are scientifically and how feasible, how easy is it to do. You require a particular transgenic animal. You require some additional viral injection, each one of this, because we want to do things under highly standardized conditions. So every time we do things off pipeline, they become much more difficult, much more expensive. Once we've selected these experiments, we carry them out using our trained technicians, a highly trained, robot, the experimental procedure, the surgery, all that pipeline. We do the recording. We do the post-recording histology, so we know exactly where we recorded. We process the data. We make the data, the metadata, all the QC, all of that is then going to be shipped all done via AWS. So you have the data. And then you guys can analyze the data. This is what happens in astronomy. And as these experiments and the instruments become more and more sophisticated, I think it's really critical that they become more and more standardized, so it's all replicable, and all the data and metadata are accessible ultimately to everyone upon publication. So that's what we're now moving towards. So in summary, the Allen Brain Observatory, we record from a large number of neurons using either optical physiology or multiphoton fluorescence microscopy or neural pixels under highly standardized condition within a high-res 3D coordinate system. All the data and metadata is freely available. We're now moving to this open scope environment, and yeah, this is what the first analysis of this what we call neural pixel survey has revealed about what we find. Once again, the vast majority of this data has not been analyzed, so people are now beginning to analyze it, a whole bunch of them, because we can just see who downloads it, and all sorts of interesting flow of information and sub-communities within these neurons that you can see there. This is our team. It's a large effort, and we thank our donor for making all of this possible. And with that, I'm done. I thank you for your attention.
Thank you very much, Christoph. This is really a very impressive work and a lot of beautiful data. And I'd like to encourage everyone to capitalize on this data, and really, I think there's a lot of fascinating computational questions that can be pursued now with this, with access to this data, and it's very laudable also that you're making all of this available to the community. So this is really very, very exciting. So there are many questions in here. I'm gonna focus on some of the questions that pertain specifically to this talk. There are also many questions about consciousness as well. I'm going to give those lower priority now. So a couple of clarification questions. Some people were wanted to know more about how you define the hierarchy in anatomical terms, particularly in circuits that have a lot of recurrent connections and horizontal connections. How do you define the anatomical hierarchy?
It's a it's a good question. We have math, as I said, 30% of all connections. You know, if you have a graph of we have 40, we have 70 nodes just in our cortex thalamus on one side of the brain, never mind the other one. We find, you know, you should find up to 70 times 70 connections. We find 30% of those. So there's massive connection. Plus, of course, there's massive connection within each area. Here we use a very simplified scheme where we classify each connection based on their pattern of postsynaptic, in which layer they classify. We impose this constraint. It's either a feed forward or a feedback connection. It's a simple scheme, so we impose this, and then we find this hierarchy. In reality, of course, we know it's much more complicated, but we simplify this. Therefore, it's rather nice, given this simplification, that we say either you are feed forward or you're feedback. Right now, we don't have a third option. It is rather satisfactory that then, if we look an average over many thousands of units, we find this nice linear correlation with the receptor field size, with the extent of simple complex, with the first time to spike, with the decoding, with the response time. But it's an approximation.
Another anatomical question here, or at least I interpret it to be anatomical. People ask whether the connectivity follows a small world network.
It depends where you look. If you just look at, yes, if you look at the entire, so people have analyzed that 2012 paper. I mean, all of that data has been online. That does follow a small world connectivity. Although, as I said, there is lots and lots of, it's sort of, yes, under certain condition you can find that. Again, it's more complex because we have 30% of all connections are present. So the small world connectivity typically is in social networks, right? When you don't have, you know, if you look at Facebook, you don't have 30% of all possible connections among 1 billion people are present, right? The adjacency, you know, if you think about the adjacency graph, it's pretty dense. But of course, the amplitude of the connection signal varies across, is log normal distribution across almost, you know, a factor of 50,000. So that just makes it more complex.
Very good. I'm reading another question here from Marco, but do I understand correctly that the neurons in the hippocampus did not respond to any of the stimuli, Gabor, natural scenes, etc.?
No, a few cells did, but not, it's a small, no, small cells, a few cells did respond, but only a very small fraction did respond to visual stimuli. Now remember, that survey was under passive condition. It might have been different, and we haven't gone back and looked in this active visual behavior. We might find some more neurons. In any case, it's a very small fraction of cells.
Very good. Another question, in the oddball paradigm, you alluded to the idea of predictive coding, but is it possible that the differences along the hierarchy can be simply explained as an accumulation of adaptation? You have more adaptation in, so you pass an adapted signal from LGN to V1, from V1 to AM, and so on, and the accumulation of adaptation gives the results that you show here.
It can't be just that, because that, remember I showed you a graph, I can pull it up again, the difference between the passive and the active, right? So in the passive condays, the animal sees exactly the same sequence, but now it doesn't have to do any behavior. So if it's purely sort of a passive sensory adaptation, and that explains a little bit, but in addition, you have this ever-growing increase. So that is not sufficient to explain the entirety of the signal. It explains some fraction of the signal.
Very good. I move on to our next question. I'm not sure I fully understand it, but I'm going to read it anyway. For the change detection task, what would be the role of the hippocampus in the hierarchy, and how does this relate to an oddball effect?
I mean, we just happen to record from the hippocampus because our electrode, you know, we're interested in the cortex and we're interested in the thalamus, right? So the hippocampus happens to be in between. We pick up all these. We haven't really done anything with the hippocampal units. We have no particular hypothesis about the hippocampus. I'm not sure the hippocampus is required for this. I guess my gut feeling is you can probably inactivate the hippocampus and the animal shouldn't have a difficulty doing this task. Yeah, and we don't make any claims about the hippocampus and the hierarchy. In that hierarchy graph, there we're just focusing on 8 areas: the 2 visual thalamic areas, LGN and the pulvinar, and 6 visual cortical areas. So we make no claim in this paper about the hippocampus.
Very good. Christoph, this is a general question about invasive experiments and neurophysiology. The question is whether inserting probes in the brain destroys tissue, and how does that affect our recordings?
Yeah, no, it's a very good question. I mean, it's very difficult to be sure, because the only way to be totally sure is to do a recording without any electrodes, and that's of course difficult to do. All we can tell, if it goes lower and to smaller and smaller diameter, and if you do post reconstruction histology, you see less and less damage. We've tried early on when we made the neural pixels, we made even thinner probes, 10 micrometer probes, but and 10 micrometer thin and 50 wide. We couldn't see any difference in terms of, for example, blood vessel pierced or dead or inflammatory tissue. So while, and these are acute experiments, these are not chronic experiments. So while we're not seeing there isn't any damage, the damage is very small. In fact, we have to add dye. We add dye eye to the electrodes, because if we don't have a dye, the damage is so little, we cannot locate the tracks of the electrode. If you have a big electrode, it's very easy to, if you do the histology, to see where the electrode was. These electrodes are so thin, if we wouldn't add the dye, we would not see the tracks. So there's probably likely to be some damage, but it's minimal.
Very good, thank you. Are there any plans to conduct developmental studies of how the anatomical as well as physiological properties change with mouse development?
No, not right now. It's a very good question, but no, we are not.
Okay. So Christoph, if you don't mind, there are a lot of questions here. I'm sorry that I cannot do justice to all of them. Can I take advantage of the fact that you're here and use the last 5 minutes to shift gears to consciousness, because there's a lot of questions about consciousness.
Sure.
So one of the questions, I think there was a discussion yesterday where I think Tommy was advocating the idea that consciousness and intelligence go together. And I know that you have strong and different opinions about this. So maybe you can say a few words about intelligence and AI and consciousness.
Yes, they're very different. Consciousness is ultimately a state of being. So consciousness I define as any experience, whether it's, just, banal like tasting a slice of cold pizza, or as rare as, having a mystical experience. All of them, dreams, memories, emotions, falling in love, being angry, being hungry, being upset about COVID. Those are all different conscious states. And so ultimately they're a state of being. It feels like something, in the words of the philosopher Thomas Nagel. Now, intelligence is very different. Intelligence is about doing. Intelligence is about acting in an intelligent way. That means you're in a particular environment and you either have to act very quickly or you have to act very slowly. Let's say you have to plan for your college, or you have to plan, you're doing your PhD, because you hope several years later you can take advantage of that knowledge gain. So ultimately, intelligence is about behavior. Now, in evolutionary structures, those things are related. In other words, we think if we look at across within species, let's say within humans, or across humans, that intelligence and consciousness is related, both at the individual level and at the species level. But they're really, I'm trying to find this picture here. You can draw a two-dimensional plane. On one axis, you can have consciousness, and you can have a measure of consciousness like phi. Some of you might know from the theory of integrated information. And on the other plane, you can have a measure like intelligence, like IQ. And in principle, they can be dissociated. And you can certainly imagine, if we think about artificial intelligence, if we think a little bit about computers, that it is certainly possible that you can have computers that will ultimately have human level intelligence, or maybe even superhuman level of intelligence, but there is no evidence whatsoever. And certainly IIT strongly argues against the notion that digital computers, for Neumann machines, will be conscious. In other words, IIT would say, yes, you can have a human intelligent, you can have super intelligence without them feeling like anything. So, but even if you don't believe that, even if you believe in the fullness of time that they can be conscious, at least conceptually, they're two very different things. Intelligence, I mean, the bottom line is intelligence is about doing, consciousness is about being.
And for people who want to know more on the particular figure that Christoph is alluding to, he presented that in a CVMM talk last year, which you can all see the video in the CVMM website. I jump quickly to a few other questions. Where does the claustrum lie in the hierarchy? Asked by Adi Nair.
We're just, okay, I don't know. We just look, it's a research project. It's an ongoing research project. The claustrum, as some of you might know, is this mysterious structure. We all have it. Humans have it. Mice have it. It's the most interconnected structure bar none anywhere in the brain. It's a thin structure underneath sort of this part, the insula, above the basal ganglia. It's within the white matter in humans. It's massively connected both ways. So it massively projects to cortex and gets input back from cortex. We don't know where it is. That's a research that we're trying to do right now.
Giambattista Parascandolo, I apologize if I mispronounce your name. He asks, are mice and humans' brains pretty much the same but at different scales? Are they very different? What can we learn about intelligence and consciousness from mice to humans?
Okay, so at the level of a cubic millimeter if I give you a cell, in fact, I've done this at my institute. I've shown people 12 cells, half of the pyramidal cells, half of them from human, half of them from mouse. I removed the scale, but people are at chance telling one from the other. Basically, the basic hardware is very similar. The genomes is very similar. Cell type, we published last year a paper in Nature where we compared human cortical cell types with mouse cell types. You can map one onto the other. They're not the same. Human cells, of course, are 3 times taller because the cortex is 3 times thicker, roughly. But they're not the same, but they're very, very similar. And the supposition is that the basic hardware is very similar. We have 1,000 times more. So, you know, we have 16 billion cortical neurons versus 14 million cortical neurons in the mouse. Overall, 86 billion neurons versus 70 million neurons. You know, the mouse fits into a sugar cube, 1 by 1 centimeter. The human brain fits into a liter, 10 by 10 by 10 centimeters. So the supposition of most of neuroscience is we precisely study mouse because they're so similar. They're not identical, but they're very similar to the structures we find in a human. So therefore, also my assumption is, yes, it also feels like something to be a mouse. It doesn't have a voice in its head like we do. It can't, you know, it doesn't worry about the weekend, about the coming weekend, but basically it is also a creature that has states of consciousness like all of us.
Thank you very much, Christoph. I'm sorry that we cannot do justice to all the questions. There are many other questions here in the Q&A, but I think that we went through some of the main ones. Christoph was kind enough to share his slides, and you have access to those slides via the web page. For those of you who are interested in Christoph's conscious side, or to be more precise, his research on consciousness, I cannot recommend enough many of his previous books, as well as the latest one, The Feeling of Life Itself, that he's pointing to right now. And then there are several previous video lectures that he was gracious enough to give both at MIT as well as in Woods Hole. So I want to thank Christoph one more time. And we're going to continue at 1:15 with Alexander Madrid. Thank you very much, Christoph.
And we're exactly on time.
Yes.
Perfect. Thank you.
Thank you very much. That's fantastic. Bye. Thank you. Thank you.